and is a HCLD. So Santiago Monet and Mark Hughes have been in this field since the field started. So Mark Hughes really was instrumental in the molecular biology of the first PGTM case. PGTM is pre-implantation genetic testing for monogenic disease. So this is looking to transfer embryos without a known disease, and that was a cystic fibrosis case. It was the first successful case in 1992. And that has gone on to be a whole new field of medicine. Santiago Monet was the first person to successfully perform pre-implantation genetic testing for aneuploidy, PGTA, and that's where you're taking an embryo biopsy and looking for the right numbers of chromosomes. And both of these gentlemen have been instrumental in the development of our field, and Santiago Monet is the last author of the paper that Manuel is going to be talking about a little bit later in this presentation. I'm just going to give you a little bit of background to what we're doing here at Cooper Surgical, because we are trying to also really innovate and bring new tests to this field. And we have a test called PGT AI, AI standing for artificial intelligence. We have huge amounts of data here at Cooper Surgical. We do a lot of PGT A cases, and we've been able to leverage that big data, terabytes of data, to use artificial intelligence and neural networks to improve the way we call this. So our calling is now purely mathematical, automated, so there's no human intervention. And we've innovated in things like establishing different truths. So where we used to use cell lines for our normalized baseline, we use actual embryos with known outcome. And our first iteration used 1,000 euploids that developed into live births or ongoing pregnancy. And we've done very large validations. This process can be kind of graphically shown here. So we would start off with an embryo biopsy, and we'd amplify it like we have done for many years, about 1 millionfold. And then we put it onto these next generation sequences, not to sequence every base pair in the embryo, but to count the number of chromosomes in that biopsy. And we used to represent this as these charts which show up and down for gain or loss, and we used to have to subjectively interpret them from the scientists within the lab. Now that digital signal is not put into an image and then reported, but stays digital and goes into our algorithms and is statistically called and then automatically put into the report and then sent to the clinic, which removes subjectivity and human errors and makes the process consistent. We've also worked very hard to try and improve the accuracy of the testing. And one of the things that we've done, and one of the major challenges of using next generation sequencing, is mosaicism. And mosaicism is something that Manuel is going to talk a little bit about in his presentation. But essentially, instead of the embryo having all cells normal, euploid, or all cells or some cells aneuploid, you have a mixture of euploid and aneuploid, a mosaic, a mixture of these. And one of the first things we did when we developed this new algorithm, PGT-AI, was to look at artificial mixtures of euploid and aneuploid cells. And this was a 270 sample experiment where we increased in purple the amount of aneuploidy in different mixtures. And you can see we can titrate down. The green line in the middle shows 20% gain or loss of chromosomes. So we can show the green line, which is euploid, from mixtures of aneuploid going up to the point where you would actually have all cells aneuploid. So this is really important because actually calling mosaics is much more difficult than calling full aneuploidies. And we need to be accurate. And we've been able to set our clinical resolution at 20%. And this is something that Manuel is going to talk about in his presentation. So just a brief summary of PGT AI. It uses automated AI calling algorithms to reduce errors. Another innovation that I haven't really had the chance to talk about, but is that we use two processes to call any change. We count the chromosomes using the next generation sequencing, and we also look at things called the single nucleotide polymorphisms, SNPs. And using that, we can detect triploidy and the origins of aneuploidies and some other things. And we've moved on to show that that has a high clinical impact and actually show benefit in the clinic using this. And we're looking to continue to innovate and bring new features to this testing in the future. So without further ado,